Couple of reminders here before we get going. One is that next week, so next Friday, so that's a little bit more than a week and a half, it's a week and 5 days, I suppose, is midterm number 1. So big reminder on that. I think what I will do is leave that for now, and I'm going to talk a little bit more about it on Friday of this week to give you some tips for studying and whatnot. And I will have some review sessions next week as well. So a bit more on that later, but just a reminder on that. The other reminder is that your Lab 3 formal report will also be due. I made a bit of a mistake. I realized just today that I had the due date for this as February 10th. So that's the day before the midterm. So what I'm going to do on the formal report is normally you get 2 weeks to do it. I'm just going to give you 3 weeks to do it. I don't want people who are working on the formal report and studying all at the same time. So what I'm going to do is give you an extra week for the formal report. So you can see the new due date here is written on the screen. And what I'm going to do is I will update that on Moodle once I get a chance. So hopefully that will help a little bit with things. I did not mean to have them all in the same week, because I know that's a lot of work to put on you. So I want to spend a couple of minutes today talking about these formal reports. And so Lab 3 is a formal report. I'm trying to remember how much it's worth. I think it's 12% of your grade. So that's a good chunk of your grade. And it's going to take a little bit of work to get this thing done. And if you take a look at the grading sheet here for the Lab 3 formal report, it's including a whole bunch of things. And so what I want to do over the next couple of weeks before class is talk a little bit about these reports. And so, today I'm going to talk about titles, for example, and another time I'll talk about introductions and methods and things like that. So last week I did send out a link to a video that talks about what's expected for the graphs and some tips on how you might do them in Microsoft Excel. So at least you have that to get started. And this is about what your report's going to look like. So if you take a look at this, you have an introduction, methods, results, discussion, etc. And each of those things are going to be a bit of volume. So most people's reports are going to be about 5 to 6 pages of written. That's double spaced. When you add your graphs in, so 3 graphs, that's maybe 3 more pages. You're probably going to have some rough data and some calculations. So when said and done, your report could be about 10 pages, give or take a little bit. So it is a pretty big piece of work. So it's not something that you can accomplish by trying to do it the night before. It's really important that you work a little bit on it here and there over the next 2 weeks. So maybe one night you tackle the introduction, the next night maybe you polish the introduction, or you work on your methods, another night you work on your results, and so on. And then eventually you put it all together, you proofread it. If you have any questions, you ask me about it, and then you have a nice polished report in the end. So hopefully this gives you an idea in terms of the what we're looking for in terms of the number of pages. I also last week sent an email with a short document called How to Write a Lab Report that kind of gives you a little bit of an idea of what's expected in each of these sections. And that really is a summary of Appendix A that's in the lab manual. Appendix A is like 20 pages long, and it really talks about these sections in a lot of detail. So do review those documents and use them as a bit of a checklist to make sure that you have everything that you need, and also use this rubric as a checklist. For example, you can see the introduction. It asks for background information, relevance of the experiment, objectives, hypothesis, etc. So make sure that you are using this as a checklist. So let me just talk a little bit about titles. That's the first thing that you'll need for your report. And so one thing that is expected of all titles in this course that you're going to make for graphs or tables or for reports, where relevant, you are going to include a species binomial name. So if you remember in week 1, we had the onion. The onion was alum sepia, I believe is the binomial name. In this case here, the binomial name is beta vulgaris. That is the binomial name for beets. So that needs to be in the title. If that's not in the title, you will be losing marks. If it's not in italics, you will be losing marks. So make sure that you get that. The title should be informative. It should say what was done. So don't just say membranes of beta vulgaris. That's not really telling me what was done, right? So here's a couple of examples, right, of some titles, maybe on a different experiment, not the one that we did, but hopefully you get the idea here. You can see it says here, which is better? Effects of cold temperatures on humans. Okay, not too bad. Tells me something about what's going on. But take a look at the second one there, right? It's giving me some information, right? So it's telling me what the variables are, effects of cold temperatures, but effects on what, right? Effects on the mental health. Okay, so it's giving me the relationship between the variables. If you think about our experiment, our variables were temperature and solvents, right? What were we testing? The membrane integrity. We were testing it on what species, and so put all those into a sentence, and you're going to get 2 marks. Like I said, don't forget the binomial name. So hopefully that's straightforward. Usually it's not too hard. Again, if you have any questions, always do ask me. I'll always look at anyone if they have a rough draft they want me to look at, or even just a piece of something. Send me an email or schedule an appointment to one of my office hours, and we can take a look at it any time. All right, so today we are going to move on to topic 5, which is prokaryotic cells, and this is my personal area of research. And background is all on bacteria, so I love to talk about bacteria. I'm going to tell you lots about them today, and the Archaea as well. So first thing, when we look at prokaryotes, here's some numbers, by the way. So some interesting numbers, 5,400. That's about how many species of mammals are on the planet. I don't know whether that seems like a big number or not, but 1,000 of species of mammals are bats, so that's kind of interesting. 10,000. So that's the number of species of microbes in about a spoonful of fertile soil. So that's a lot, right? Just one scoop of soil, and you got about 10,000 species, and probably more organisms in that scoop of soil than there are people who have ever lived. 4,000. So that's about the number of species of prokaryotes that have been characterized, and we think there's probably maybe 4 million. We don't know. It's hard to characterize them all. It's probably going to take us a long time for us to do that. Some of them we can't grow in the lab, so we can't characterize them, but we know they're there because we can see them under a microscope, or we can detect their DNA. So there are a lot of things out there, a lot of very small things. Another way to look at them is the numbers just around the human body. So the human body, about 30 to 40 trillion cells. About 10 times as many bacteria in and on your body. So that's kind of incredible if you think about it in terms of, you are more bacteria than you are human in the number of cells. By mass, you're more human. Don't get that wrong. So one other thing to say about these is these things are everywhere. They're in the soil, they're in the water, they're in the air, and they include both the bacteria and the archaea. And the prokaryotes, as we talked about in week 1, prokaryote means before nucleus. So that means they have no nucleus. They're considered to be more primitive than eukaryotes. And we'll talk about eukaryotes in topic 6. So before we get any further, here's our picture. Remember, we showed this before, showing our eukaryotic cell and our prokaryotic cell. Like I said, the biggest difference is the fact that the prokaryotic cell has no nucleus, and the eukaryotic cell has a nucleus. So what I want to do is, I just got to pull up a note here and make a few notes about prokaryotes versus eukaryotes. And so just hold on a second. I just got to find the document. I thought I had it right here. So this is something that I usually do on the whiteboard again, but I'm going to do it in a Word document instead, and come back to some of these concepts a few times. So there it is. Okay, let me share this with you. There we go. Okay, so like I said, let's just make a little table here comparing prokaryotes and eukaryotes. Okay, so for starters, the first section you can see, it says includes what? So in this case, we're talking about the bacteria and the archaea for the prokaryotes. Eukaryotes, this includes animals, plants, fungi, protists, and sometimes we consider algae their own thing. So I'll throw that one in there. Okay, so that's hopefully pretty straightforward. Maybe the new one for some people will be the Archaea, and we're going to talk about those shortly. What's the typical size of a prokaryote? So 1 to 10 micrometers. So I'll get the special symbol for micro. Yeah, it's right there. 1 to 10 micrometers, so very, very tiny. Eukaryotes, 200 on the small end, and in some cases up to 500 micrometers. And some are much larger, in fact. In the human body, you have a muscle cell that goes from your hip all the way to the end of your toe. So depending how tall you are, that muscle, that cell is like, maybe up to a meter long in some people. There's always exceptions. This is, of course, biology. So the next couple of questions are just yes or no type questions, right? Membrane-bound nucleus, no, and yes, because of course eukaryotes, remember U means true nucleus, true nucleus. So that is what to remember. Other membrane-bound organelles for prokaryotes, no, and eukaryotes, yes. And you might remember a lot of the organelles that you talked about in high school. We've got mitochondria, we've got endoplasmic reticulum, lysosomes. We could go on for a while, but we will actually in the next unit. So let's just stop there for now. What about cell walls? We've already talked about cell walls. Okay, so we talked about bacteria, of course, have cell walls, and the cell walls are made out of peptidoglycan. The Archaea, they sometimes have pseudopeptidoglycan, and some have S layers. Okay, and the eukaryotes, we talked about them as well. Remember, plants have cellulose. And what else did we talk about? We talked about fungi. The fungi have, let's try that again. Fungi have chitin. What else did we talk about? Protists. We'll just say big variety or large variety. Some of them don't have a cell wall. Some of them have weird things. Some of them have cellulose. Some of them have unique carbohydrates. So we're not going to get into all the protists. But what about animals? So remember, the animals have no cell wall. But you could put in your disclaimer that some animal cells have an extracellular matrix. Okay, so there's a few other things down here. I'm going to add some details too. And like I said, I'll probably come back to some of these concepts later on in the course. This is a big theme of Biology 107 is the differences between the different cell types, the prokaryotes and the eukaryotes, etc. So like I said, I'll probably come back to some of these concepts. What can we say about the DNA? There's quite a bit we could say about the DNA. I'm just going to say a few kind of minor things here. One is that in prokaryotes, they have a circular chromosome. So maybe I will just put chromosome, and there's a couple of things I'm going to say about the chromosome. The chromosome is circular, usually only one. So by the way, most of these things you could put the word usually in front of. There are a few prokaryotes with linear chromosomes. There's one or two that have more than one chromosome, but usually one, right? So we'll just say one. What else was I going to say about it? Maybe that's enough for now. Like I said, we'll get into DNA later on in the course. And so for eukaryotes, we have linear chromosomes, meaning it's kind of like a piece of string with a beginning and an end. Often many. You could be diploid or haploid or other things, okay? And one other thing to say is that the DNA is packaged with something called histone proteins. And we haven't talked about those yet. And it's going to be a little while later we'll talk about them. But just wanted to throw that down so that you've heard that word, and you'll hear it again later on in the semester. Somebody has a question that says, what does ECM stand for? Extracellular matrix. So I can fill that in if you like. Extracellular matrix. So that's like your collagen or your bone structure, those kind of things. Okay, what else do we have? Oh, 3 more things. Okay, what about ribosomes? Yes, they have ribosomes, and we'll just say that the prokaryotic ones are smaller, and eukaryotes have ribosomes, and they are larger. Technically, again, this is a detail that we're going to talk about eventually here, maybe even today, is the size is 70S and 80S, and we don't know what that means yet, so we'll get there. Reproduction. Okay, so maybe I'll do the eukaryotes first. You probably know that eukaryotes do mitosis and meiosis, okay? And prokaryotes do something called binary fission, which is kind of a funny word. It's sort of overly technical, and it basically means cutting in half. So we'll get to that shortly as well. So don't get this mixed up. Sometimes people seem to want to say prokaryotes do mitosis and eukaryotes do meiosis. If you were doing meiosis all the time, your body wouldn't be functioning properly. It's really only your sex cells that do meiosis. All right, and then the last one there, motility. Maybe I'm not going to fill that one out for now. We're going to get to that eventually, but I won't worry about that one for now. I think that's enough. Kind of some of the main differences. So I'm just going to scroll up and throw out a couple highlights. So remember, this is a big one, right? Who has a nucleus? The eukaryotes have a nucleus, okay? And then everything else is kind of all the, the details where the devil is found. Like I said, we talked about cell walls. We're going to talk about organelles. We're going to talk about DNA. We're going to talk about ribosomes. We're going to talk about reproduction. And so hopefully all those things will start to make more sense as we get into those details. Okay, I feel like there was something else I was going to say here. Let me just check my notes for a second here. Oh yes, okay, we're going to get to Archaea here in a moment, okay? But let me just go back to the PowerPoints. How do I get back to PowerPoint? There we go, back to the PowerPoint. So 2 kinds of cells, the eukaryotes, that includes animals, plants, and fungi, and a bunch of other things like protists, and the prokaryotic cells, they include the bacteria and the archaea, okay? And so let's talk about those a little bit more. Technically, most people divide life into 3 groups. The eukaryotes, these of course have the nucleus, and the bottom 2 groups, the archaea and the bacteria. And some scientists, disagree on which ones are more closely related. I've seen schemes where the archaea are closer related to the eukaryotes. I've seen schemes where the archaea are more closely related to the bacteria, and not really my area of research, but they are 3 different categories. And I do want to talk about the archaea in a minute as to why they're different from the bacteria. They have no nucleus, but there's a lot of differences here. And I'll get to that in a second. I think I have a slide or 2 to share first. Oh, here's one of the slides. So one thing that was noticed, I think this is back in the 1960s or 70s, if you go to one of these places where there's a hot spring, and you can, the water's very hot. In some cases, it's almost boiling, which is, people thought that was enough to kill all life. And they would stick a microscope slide in there and they would see organisms on it. And it was like, wow, what is this? And so they realized they were dealing with something unique. And turns out that these organisms were unique and different from what we have now. Not all archaea are weird and unique, living in extreme locations. Some are living in more, how shall we say it, natural locations. Some of them produce methane, for example, so they're living in your intestinal tract and they're producing intestinal gas, which is, embarrassing at times, but that's just the way things are. This is life. And sometimes there's weird and wonderful things. So, like I said, I want to make a few notes about the archaea here. And unfortunately, I'm probably not going to come back to them again. Maybe here and there I'll mention them, but mostly when we're talking about prokaryotes, we're mostly talking about bacteria for the most part. And maybe that'll, there's some reasons for that I will maybe highlight here in a moment. So hold on, did I get back to the Word document? Yes, I did. Okay, so back to the Word document. Some notes on the Archaea. What is going on here with my, there we go, okay. Some notes on the Archaea. Don't know why this is centering. Let me just slide this back over there. There we go, okay. So these things are prokaryotes, right? We've just been talking about that, and that means no nucleus. Okay, that's the first thing to know about Archaea. And they are a unique domain separate from the bacteria. Okay, so what is unique about them? So remember, they have unique phospholipids. We just talked about that last topic. Phospholipids. Some of them are branched phospholipids, which is kind of a unique biochemistry. Some of them form those weird monolayers, but it's just enough to say that they have unique phospholipids. Some live in extreme environments, like hot springs. They are found almost everywhere. So soil, found in your intestine, pretty much wherever bacteria are found. We could say some are methanogens. Methanogens, that means they produce methane. And the other thing we can say, without getting into all the particularities of them, but they have they possess many unique, we'll call it biochemical properties, right? So for example, we talked about pseudopeptidoglycan, in the cell wall. They have unique RNA polymerases. We could go on in terms of the biochemical things. And if you don't know anything about RNA polymerases, it's not going to make much sense to you. So kind of this is really the key point, right? Is they possess many unique biochemical properties, things that are not found in the eukaryotes or in the other bacteria. So that's kind of one of the big things I'm going to say about the Archaea. There is one other thing I want to say about the Archaea. Oh, what happened to my notes here? Let's just get that back over there. Is that many are very slow and hard to grow in a lab. So that's why we know less about them and we talk less about them. They're kind of laborious to research. And maybe the last thing to mention about these things is that there are no known pathogens. So that means that none of them make us sick, that we know of anyway. And so this is one reason why we don't talk about them as much and we don't research them as much as well, because of course, if they're not having effects on our health, then maybe we're less interested. Not that they're not interesting, it's just something that, makes less money available to do them, and they're hard to grow in a lab. And so, you only need a few reasons to why not look at something. So that, like I said, is kind of mostly what I'm going to say about the Archaea. Mostly everything else I'm going to talk about in this unit is more or less referring to the bacteria from a general sense. But that's kind of what I'm going to say about them. I don't know when somebody put this question up here about how do they differ from the bacteria. All of these ways, right? Unique phospholipids, they live in extreme environments, they produce methane, some of them, some bacteria don't produce methane. The cell wall is different, RNA polymerases are different. There's a lot of things we could get to on a biochemical level in terms of how they're different. But like I said, this is one of the big ones. They're not making us sick, whereas there are bacterial species that do. All right, so back to the PowerPoint. Let's kind of get into some of the details here. So there's a note there. It says, they're found all over the biosphere. Doesn't mean they're not interesting. Just means we know a lot less about them, unfortunately. Okay. Oh, I guess I was going to talk about results. We'll get there next day. That was probably from last semester. Let's talk about bacteria here for a few minutes, and we'll get into some of the details in terms of their cellular biology. So what do bacteria do? Probably most people know a little bit about bacteria, but it's worth it to kind of highlight some of their features in the biosphere. So many of them live with us symbiotically. So this is E. coli. E. coli lives in your gut, and it is making vitamin K, it's making folic acid. It's a symbiotic relationship. We're giving E. coli food in a warm, moist environment. It's preventing some pathogens from living there. Now, some E. coli might make us sick, and we'll kind of address that in next lecture, because it's not fair to say E. coli is always making us sick. In fact, most of the time, E. coli is beneficial to human health. Some bacteria do cause nasty diseases. This is the one that causes the plague. It's called Yersinia pestis and was very famous for killing millions of people in the Middle Ages in the 13 or 1400s. And lots of things could be said about it as well. Many bacteria are photosynthetic. So they're making oxygen for us, which is kind of awesome. Here's cyanobacteria, probably making at least half the oxygen on the planet out there in the oceans. Many are involved in all sorts of areas of nutrient cycling and parts of the biosphere. These ones are found in the soil, and they fix nitrogen and basically fertilize the soil for certain plants. Like I said, many roles out there in the biosphere. We could go on and on. We could talk about bacteria in the gut of a cow. We could talk about yogurt making, and some of these things we'll touch on perhaps over this lecture. What we are going to talk about, though, is a little bit more of the cellular biology of these prokaryotes. And you can see here's kind of a picture of one. And there's no nucleus. Remember, no nucleus in a prokaryote. But it does have a lot of other features that are worth talking about. So let's get into those. And what I am going to do is, as I'm talking about some of the features, what I want to do is also make some notes. Okay. So just giving you a heads up on that, as we are going to make some notes on bacterial structures. So maybe make yourself start making yourself a table or something like that. So just give me a second here. I'm looking at my own notes and making sure I have everything here. Don't want to forget anything. All right. So first place to start is, let's talk about the cytoplasm. So the cytoplasm is the inside of the cell. And a lot of people like to think of it as just water, but it's not. And this is why I'm showing you this painting done by this artist. And he's trying to show that the cytoplasm is full of stuff. It's full of RNA, it's full of ribosomes, it's full of enzymes. So let's talk about the ribosomes for a minute, and I'll come back to the cytoplasm. So here is the difference between the prokaryotic and the eukaryotic ribosomes. So ribosomes are made out of both RNA, so you can see in the list here, RNA and proteins. And this is trying to show the composition of these things. And so the prokaryotic ribosomes are a little bit smaller. They are 70S, so that's a measurement. It's actually, S stands for Svedberg. Svedberg was a Swedish guy who was studying ribosomes, and he was looking at how fast they sediment in a centrifuge, and so they named the unit after him. So 70 is smaller than 80. So eukaryotic ribosomes are larger than prokaryotic ribosomes. So maybe some people are trying to remember, what does a ribosome do? A ribosome is the site of translation. So we'll talk about that later on in the semester too. So a bit more on the cytoplasm, and then I'll write a couple of notes for you guys. Just want to show you what else is in this picture. The cytoplasm is loaded with enzymes. The DNA is found there. And there's a protein, DNA polymerase making the DNA. You can see one strand going in and two strands coming out. And so we'll come back to DNA in a moment. So what I'm going to do is make a couple of notes on those things. And like I said, some of these things we will come back to later on in the semester, right? Okay, so Oh, maybe I got to share this. Sorry about that. Oh, no, it is sharing. Okay. The cytoplasm. So what is the cytoplasm? Cytoplasm is water, of course, and there are solutes there, and there are enzymes. So this is the area within the plasma membrane, which really just means cell membrane. It contains the cell's contents. So including the solutes and the enzymes and things like that. What are ribosomes made of? Ribosomes are made out of ribosomal RNAs, that's what the little r is for, plus proteins. And what do ribosomes do? Ribosomes are the site of translation. So translation is protein synthesis. And this is from a messenger RNA sequence. So like I said, we're going to talk a lot about translation later on in the semester. We have a whole unit on translation. So what are enzymes? Enzymes are made of protein. And these are catalysts. So they speed up chemical reactions. Okay, so I'm hoping there's no surprises there. If I am going to clap fast, please do ask questions and slow me down. I'm trying to go faster on the areas I'm hoping you know, and slower on the areas that are new to you. Okay. If you don't have all this, I will come back to this this note in a moment. You may notice there's a bunch of blanks there that will get filled in here shortly. So there's our cytoplasm. Like I said, a whole bunch of things in there to think about. And let's think about the DNA here for a minute, okay? We were talking about the DNA in a bacteria or an archaeal cell as being circular, right? So you can sort of think of it as a big circle, but it's really not quite that way. The chromosome, rather than being a big circle, is sort of, it's more like this. It's more packaged up a lot tighter. But it does join on itself, and then you have a chromosome. You can see in that image there on the right, this here is a chromosome here. I'm going to put a big X star on it. That's a chromosome. And this cell here is actually dividing. So it actually has two chromosomes at the moment because it's replicated its DNA. And so you can actually see two chromosomes. And there is no nucleus, but the chromosomes do show up on an electron micrograph. So because of that, these people back in the day who were doing electron micrographs, they started giving it a name and they started calling it the nucleoid. So don't get confused, because a nucleoid is not a nucleus. It just means this is where the DNA is and it stains really well on an electron micrograph. Now, most bacteria have one chromosome, but a lot of them also sometimes have what you could think of as a kind of a mini chromosome. And these mini chromosomes are something called a plasmid. So normal chromosome, you're looking at, well, in the case of E. coli, maybe around 4,400 genes. A plasmid, you're looking at, anywhere from 1 to 15 genes, maybe 3 is more common. So it's very, very small. And it's helpful though. Sometimes a few extra genes can mean you're going to survive, right? So I'll show you some examples of genes that are coded on plasmids. This is the big one that we are concerned about in the medical settings, is antibiotic resistance. Plasmids can code for genes that can make organisms resistant to drugs. So this is a really big deal. And sometimes these plasmids can actually get shared between bacteria, which is kind of scary. There's other things on here that we haven't really talked about: synthesis of pilus, toxins. And in many cases, basically what this means is the organism is more pathogenic. It can make people sicker. It has a better ability to be a pathogen, which is why we're really concerned about plasmids. I think I have another picture of a plasmid. No, I don't. But I do have a picture of binary fission here. So let me just go back to this previous slide for a second. As I told you, this one here is in the middle of dividing. And so I just want to talk about that for a moment, and then I'll make some notes on the chromosomes here. So the process of cell division in bacteria is not mitosis or meiosis, it's called binary fission. So binary means two, fission means cutting in half. So kind of what this means is we're cutting the cells in half and we're making two cells. I don't know why we have to have a technical term for this, why we can't just call it cell division, but this is the way I didn't name it. That's sort of part of the problem, right? I would name things differently. So take a look, we have a cell up here. And the cell has that chromosome, and the chromosome is going to replicate. And so you can see they're showing it starting to replicate here. And notice what is happening is that the chromosome, it's actually attached to the membrane here. So when we do division in bacteria, we don't need spindle fibers like mitosis. In fact, we can divide, and as the cell stretches out, it literally pulls apart those two chromosomes, right? This cell is growing, and one of them is attached to the left, one of them is attached to the right, and it pulls chromosomes away. And then in the end, we get our cells divided. And they look like that. Sorry about that. I didn't really want the volume to go on that video. There's a couple of videos there showing bacterial cell division. So you can see them on the top one, the E. coli is stretching out, divides, stretches, divides, stretches, divides. It doesn't happen that fast. It takes about 20 to 30 minutes for E. coli. There's another organism on the bottom. I'll press play on that. You can see this organism has a different shape to it. It's more of a circular coccus shape, and it's kind of doing the same thing, stretching, dividing, stretching, dividing, stretching, dividing, and so on. Hold on a second. So just for comparison, right? Mitosis, very different, right? Remember mitosis, you may remember we have, let's see here, what do we have? We have prophase here. This is metaphase, anaphase, telophase, and so on. Okay, so much more on that in topic maybe 12. I think we're talking about mitosis. So binary fission, much simpler, no spindle fibers. There's one chromosome to deal with that's replicated, and you know, you get two daughter cells. So much simpler process. So before I do the notes, couple of quick test yourself questions. Let's take a look at them. True or false? Bacteria divide by binary fission and archaea divide by mitosis. So that is false. Does archaea also divide by binary fission? Number two, the nucleoid region is surrounded by a membrane. That is also false. No, it is not. Remember, the nucleus is surrounded by a membrane, and the nucleoid region is not. Number three, prokaryotic ribosomes contain 60S and 40S subunits. I know everyone's thinking, oh no, what does that even mean? I'm just going to go back to that slide to show you what that is talking about. So sometimes on a test, I might ask you some specific questions. This really is to reward those people who study really hard. So let me just go back to that slide here. It's not too far back. Oh, there it is right there. So it turns out, I guess I forgot to mention this, right? That there's two subunits in a ribosome. This is called the large subunit here. This is the large one. And this is called the small subunit. And so overall, the prokaryotes have a 50S large subunit and a 30S small subunit, whereas eukaryotes have a 60S and a 40S. So it's a bigger structure. So I'll go back to that question, which is right here. And so the answer is false. This is really referring to eukaryotes on this one here. Okay, so I'm going to go back to that Word document and fill in a couple of the notes here, right? So we have the nucleoid. Okay, so remember the nucleoid is basically just DNA. And what's its function? Well, what's the function of DNA? DNA stores the genetic material of the cell. Plasmids are also made out of DNA. Carries a few genes. So a few genes meaning, you know, typically, like I said, 3 to 15. Could be a few more, could be only one gene, but that's kind of the typical range of what a plasmid is. Maybe I will add and just say this is circular DNA. How about that? Because of course that would be true. Okay, so we have a few other things to talk about. We're going to talk about the cell wall, but maybe I would fill that one in since we know the answer already. We know the cell wall. I don't know I have a space, by the way. Don't worry about the space. We know that the cell wall is made out of peptidoglycan. We've already talked about that. What does a cell wall do? So it provides support, and it also provides shape to the cell. Maybe I could say it provides protective or support and protection. There we go, we'll put protection just in there like that. So it's a tough structure, right? And that's what it's going to do. All right, so I'm going to flip back to the PowerPoint, and we're going to talk about the cell wall, and we probably won't get to any of those other things today. So we really want to talk about the cell wall. So let's talk about the cell wall. The cell wall is shown in this picture here. If you take a look, we have two membranes. So this is E. coli. E. coli is gram negative. Remember, we talked about gram negatives already, and we'll talk about it again in a minute. Excuse me, just something got in my throat there. So sometimes we usually call the main membrane the plasma membrane. So I'll call it the plasma membrane there. We have peptidoglycan, which is the carbohydrate peptide structure, and we have the outer membrane. So here's a different picture than what I showed you, or maybe it's the same picture. I can't remember if this is the one I showed you when we were talking about the cell walls. But we have gram negatives and we have gram positives. So the gram positives on the left, they have a thick peptidoglycan layer and just one membrane. The gram negatives on the right, they have two membranes, a plasma membrane and an outer membrane, with a peptidoglycan sandwich in the middle. So the gram positives stain purple and the gram negatives stain pink. And that's what's shown in that little micrograph in the middle. And this is actually what we're going to get a chance to do in this week's lab, is we're going to do a gram stain and see the differences between the two different types of species. So I just want to bring you through the procedure of the gram stain and take a look at how this whole thing works. So first of all, you can see you've got these cells here, and they're not very visible, right? So the first thing you do is you add this crystal violet. And the crystal violet you probably know is purple, right? And so it kind of just sticks to everything initially anyway. And so at this point, everything's going to look purple. So that's not good, because what we want to do is distinguish the differences between the two types of cells. So what you do is you add this thing called a mordant. A mordant means a fixative, and we actually add iodine. So how does that work? So the crystal violet, by the way, so here's my crystal violet, right? And it's positively charged. So here's the crystal violet. And if you put in iodine, iodine is negatively charged. So what it does is it kind of just binds to the crystal violet. And so now what you have is not just crystal violet, but you have these large complexes. And these large complexes get stuck inside the gram positive cell wall. The gram negative cells, they're not really stuck. They're kind of just loosely stuck on the outer membrane at this point. So the next step, what we do is we wash everything with ethanol or acetone. And what that does is it washes away the excess crystal violet and actually washes away some of the outer membrane on the gram negatives. And so they end up becoming decolorized, which means they lose their color. So the last step, you throw in another stain called safranin. Sometimes people call it safranin red because it has a reddish pinkish color. And so what that is going to do is color everything. Now here's the thing, the gram positive cells are purple. They're a nice dark purple. And so if you mix pink and purple, you still get dark purple. You can't see the pink at all. Whereas the colorless gram negative cells turn out pink. And that's kind of what they look like there. So anyway, you'll get a chance to do this in the lab, all 4 steps, and I'll do a demonstration of it on Thursday. Here's some micrographs of some of these cells. And this can be used in many cases to actually identify the actual cells. You take a look on the left there, you can see 3 different types, and they look very different under a microscope. Staphylococcus, we've talked about a little bit. Bacillus cereus, we're going to be staining that one in the lab on Thursday. We're not going to be staining anthrax, so no pathogens in our lab. On the right, you can see some different organisms there. We will be staining E. coli. We will not be staining gonorrhea. We're not going to have any pathogens in the lab, but you can certainly see the differences, and it can help you to identify the organisms. One other reminder that we talked about last day is that the gram-negatives have a couple of unique things, and this is just the same slide I showed you last day, just as a reminder that the gram-negative cell wall is unique, has unique lipids in its outer membrane, and these are called lipopolysaccharides, and those are actually an endotoxin, and if you get those in your blood, if you have a blood infection, it can lead to a very severe immune reaction. You don't want that. And we also have the periplasm, and this is the region between the membranes, and this is where some enzymes are found, and sometimes they're involved in drug resistance, so this is an area of interest by some people. One other thing to mention is that gram-negatives and gram-positives, they don't represent every type of bacteria. There are a few other types. And we'll be asking you about this one here, but it is something that you may have heard of. You may have heard of tuberculosis. Tuberculosis is a very bad lung infection. You don't want it. And its membrane is actually a little bit different. It has a little bit of peptidoglycan in there. You can see that's listed here. But it has a lot of these other fancy carbohydrates and fats. And in fact, this one here, the mycolic acids, that's very waxy. And so it's got a very different biology because it has a different cell wall. And so sometimes people call this an acid-fast cell wall or a mycobacterial cell wall. But just a little tidbit for you, just to show you that something, that there are a few different types of organisms out there. There's a stain of Mycobacterium tuberculosis. You can't actually gram stain this organism because of this waxy substance. It doesn't allow the stains to get inside the cell. All right, I'm just doing a time check here. And we've got 2 minutes, which I think is just enough time to do this little matching kind of thing here, right? So let's take a look at this here. I've got a bunch of different types of cells on the left. I've got a bunch of information about cell walls on the right. And so let's work down this list here and see if we can figure it out. I intentionally made it a little bit cryptic to get you people to think a little bit here. So if you take a look at this here, right? Chitin, hopefully that's an easy one. Chitin belongs to fungi. Yeah, that's the only time that you're going to see chitin, although you might see it in some weird protists or something like that. The next one, never peptidoglycan. So I know that one's really cryptic, but what I was trying to do was say, hey, remember that archaea are different from bacteria. They don't have peptidoglycan. They're not bacteria. They might have pseudopeptidoglycan. They might have an S layer, but they never have peptidoglycan. 2 membranes. Hey, this is easy. Everything's in order. Gram-negatives. Remember, gram-negatives are the sandwich membrane cell wall thing with the outer layer. So there you go. That's an easy one. Next one, may have an extracellular matrix. Okay, I guess we gotta go up here to the animal cells now. All right, that's my ECM, extracellular matrix. Okay. All right, what else do we have here? Stains purple and animals cannot digest this. So hopefully you remember just from the previous slides that gram-positive bacteria do stain purple. And that of course leads the plant cell wall, which the plant cell wall is made of cellulose, and animals cannot digest cellulose. Cellulose is fiber, it's indigestible by animals. You may have bacteria in your gut that can digest it, but we can't digest it on our own. Okay, it looks like it's 4:50 now. I guess we'll wrap everything up and we'll come back and finish off these notes next day. And on Friday, we'll start topic 6, and I will also talk about the midterm on Friday as well.